Hello, welcome to another video from the conscientious biologist, Ben Gallagher. This is a really important one, and whilst it's from the GCSE syllabus, this is the kind of topic that everyone ought to have in their general knowledge because it's about your health and it's about keeping you well. This is the fifth one in the disease playlist, and this looks at white blood cells, which are the soldiers, the main element that fights for you in your immune system. As I've just said, it's from the GCSE syllabus, but it is important to everyone, so do have a watch and head over to my YouTube channel and please do subscribe if you haven't done so already. Thank you. So in previous videos, we've looked at non-communicable and communicable disease, and we've seen how communicable disease is spread by pathogens, which move from person to person, infect your body and make you ill. In the last video, we looked at barriers to infection, which is ways that your body tries to stop those pathogens from getting into you in the first place. Things like your skin, stomach acid, mucus, things like that. What this video focuses on is if those pathogens do get into your body, how does your body fight back and fight them off? Now, this is done by the white blood cells, which are the body's soldiers. Now, what you need to remember is that we are at war. Pathogens are all around us and they're constantly trying to attack us. It's not that your white blood cells and your immune system activate only when you're ill. Battles in this war are fought every single hour of every day, and that's against pathogens. Pathogens are the enemy. So it's not just when you're ill. Right now, there will be pathogens floating around in the air that are trying to get into my body. My barriers should stop them. But if some of them do, my white blood cells should sort them out and hopefully kill them before they have a chance to make me ill. This war is constantly raging. Now, you've got loads of different types of white blood cell, but there's two main types that you need to know about for GCSE, and really it's two main types that do most of the work. You've got phagocytes and you've got lymphocytes. Now, we're going to look at each of those individually on a slide of their own in a moment, but what we need to do first is look at some really essential background knowledge so that you can understand how lymphocytes and phagocytes do their job and keep you well. We need to look at this concept of antigens. Now, an antigen is like a signpost stuck on the membrane of a cell to identify it. So think about all the work that we've done on cells previously. You've got things like this. You can see the membrane, the cytoplasm within it. And on this cell, you'll see that I've stuck a load of little sort of triangles sticking out of the surface. Those would be the antigens. Those would be like this signpost here. They would mean something, but instead of actually saying Ben's cell on them, it's the shape of these things that represent what they are. Now, antigens are proteins. And remember, the most important thing I've taught you, proteins do everything. All a cell needs to be able to do to be metabolically active and classed as living is produce its proteins by using the ribosomes to read the DNA to build the proteins. Now, the thing about proteins that makes them unique, that gives them their function, is their shape. Same with antigens. It's the shape of the antigen protein that makes it unique. So instead of having writing on it like that one that says Ben's cell, it would be a shape that means Ben's cell. Okay, that shape is recognized by other things. Remember, language, those words Ben's cell, that's just a code that we recognize because we've learned to read. Well, your cells have learned to read the shape of proteins instead of the shape of letters. Now, cells can have multiple antigens signposting different things about them. So whilst these triangular ones might mean Ben's cell, might mean this cell belongs to Ben Gallagher, it's part of Ben Gallagher's body, there might be other antigens that say, well, it's a pancreatic cell. Another one that says specifically it's a beta cell from the islets of Langerhans within the pancreas. So there's lots of little antigens all over its surface that identify exactly what that cell is to other parts of your body. Now, the really key thing about antigens as far as it fits into immunity is that all of your cells, every single one, will have one specific antigen that's unique to you. And we call that a self-antigen. So every cell in my body will have a protein on its surface of a specific shape that's pretty much unique to me. I'm the only person in the world, possibly, who has that specific antigen. And it means Ben's cell. It means this cell belongs to Ben Gallagher. That's what that antigen means. Now, this is where it makes life easier for your white blood cells, because your immune system, which is your white blood cells, uses the self-antigens to tell what cells belong to you. And if it doesn't have a self-antigen, of course, it could be a potential pathogen. So your body uses antigens to work out what cells belong to you and what cells don't, because if it doesn't belong to you, it's possibly going to be a pathogen that could do you harm. Okay, let's look at the first one of the two main cell types. These are awesome. These are one of my favourite cell types. And I know that sounds extremely geeky that I've got a favourite cell type, but I don't care, I am geeking. Phagocytes are awesome. Phagocytes basically have one mission. It's to search and destroy. They only have one purpose. It's to find and destroy anything that they don't recognise as belonging to your body. And remember, we've just talked about antigens. Antigens are the things that show that this belongs to the body. If a phagocyte finds a cell and doesn't recognise it, in other words, it hasn't got a self-antigen, that cell's going to be in trouble. What they're doing is just checking for the presence of self-antigens, and they're doing it constantly. They are all the time moving around your body in the blood, checking every cell they come across. But as well as moving around in your blood, they can leave the blood and they can go deep into the body tissues to literally search every single bit of you. Now, what they have to do, of course, is check every cell. So it's a very time-consuming process, which is why they're constantly doing it. Let's look at the mechanism by which they do that, and let's look at what a phagocyte looks like. So they look a bit like this. Now, that hasn't really got much of a shape. And if this was like a live video of that phagocyte, you'd see it kind of pulsing, and a bit of it, you can see how the membrane kind of is pushed out in blobs. Those blobs can push forwards and back and move around. Phagocytes look and move like they're unicellular organisms. They can drag themselves around. They push out a bit of their membrane like a leg and grab on and drag along by it. They can reach out and grab hold of things. They're amazing things to watch through a microscope. And it's because of that very gelatinous, moldable shape that they can do their job and check what cells have got. Now, weird thing about some types of phagocytes, but they're the type that quite often are used in your exam in pictures, is they have a really weird nucleus that looks like this. It's called a trilobed nucleus. Still serves the same function. It still houses the DNA. It just looks a bit different, which is quite helpful for you in identifying phagocytes, because generally in books and in the exam papers, this is how they draw phagocytes. Okay. Now, what a phagocyte does, of course, is it looks for the self-antigen. So if here's a self cell with self-antigens, the phagocyte will go up to it and it will press its whole body around the self cell and feel for the antigens. Now, how it does that is that a phagocyte has lots of protein receptors in its own membrane, and it's feeling to see if your antigens will lock into one of its own receptors. It's kind of like the active site on an enzyme. It will be complementary. Now, if when the phagocyte presses onto it, if it can't lock in with a self-antigen, because there are no self-antigens, it knows that doesn't belong. But on this cell here, it's happy with that. It can give it a tick and it moves along. And that will happen again and again and again as that phagocyte just checks cell after cell after cell, pressing onto it, locking in with a self-antigen, going, oh yeah, that's got one, that belongs, release, move on to another cell. However, every now and again, there will be a pathogen in your body. Now, if there is, the phagocyte will move onto it, will press onto it just like before, but now the protein receptors on the phagocyte haven't got anything to lock into. So they're going, well, there's no self-antigens here. I don't recognize this cell. And if it doesn't recognize it, it continues doing what I've tried to show on the picture here, of pressing itself right the way around the pathogen and swallowing it. Now, once it's swallowed that pathogen, it uses digestive enzymes to destroy it, to literally digest and break that pathogen into pieces. So it's searched for it, and now it's destroyed it. This is what pathogens do. Now, that process here, that swallowing and digesting, is called phagocytosis, because it's done by phagocytes. And phagocytosis is just when a phagocyte doesn't recognize a cell, so that means that cell must be one without a self-antigen. It engulfs it, that's the swallowing, and destroys it using digestive enzymes. You need to know that word phagocytosis, that it's done by phagocytes, and that's how phagocytes serve their purpose. So really amazing cells. Take a screenshot of this one because this is key information about how phagocytes work. And this is constantly happening in your body. Right now, hundreds, if not thousands of phagocytes are in your body checking your cells, finding pathogens and destroying them before those pathogens can do you any harm. Okay, second type of white blood cell. These are lymphocytes. Now, there's a lot of information on this slide, and it's really, really important because it's lymphocytes that are going to lead us on to understanding what immunity actually means. So lymphocytes, these produce antibodies. Let's have a quick look at what antibodies are. So an antibody, like anything that does something, is a protein. Remember, proteins do everything. If antibodies are going to do something in a cell, they probably are proteins. So these are proteins. So again, it's their shape that determines their function. And this is kind of what an antibody looks like. What you've got here are two very clear regions. You've got this top one, which is variable, means it can differ from antibody to antibody. And you've got this bottom section here, which is the constant part. All of your antibodies, every single antibody you produce from no matter what lymphocyte, they all have identical constant parts. Okay. Now, variable part will be complementary in shape to an antigen on a specific pathogen. So if I pull up these pathogens again, I know they're just cartoons. Pathogens don't actually look like that, but it serves our purpose because if you look at them, they've all got different shapes on the outside. Some of them have got eyes bulging out, some of them have got arms, some of them have got tentacles. They all look different on their surface because pathogens also have antigens. Literally all cells on the planet, pretty much, have antigens that are unique to them. We have our self-antigens on their cells, but pathogens will still have their own antigens. That's like all the various bits sticking out of the structures on here. Now, the variable part will fit an antigen on a specific pathogen. And try and remember it this way, it fits onto the body of these pathogens. So an antibody is something that's going to do harm to the body of the pathogen. Okay, all these little guys, there, 6 of them there, the 6 bodies. Antibody binds to the body of the pathogen. Now that's really important because another thing the variable part can bind to is the pathogen's toxins. If you look at these pictures now, I've put these little star shapes. Those are toxins, poisons basically, that pathogens can release that can make you ill. If the variable group is complementary and will fit onto the toxin, then you don't call the antibody an antibody because it's not binding to the body of the pathogen, it's binding to their toxin. You can probably guess this. We call it an antitoxin. So I've changed the title in this box above me now to antibodies and antitoxins, but that variable bit binds onto those. If you haven't understood that, don't worry, it'll be much more clear when I put a load of information in that white space over there. Now that constant part though, that's always the same no matter which lymphocyte made it. Now each lymphocyte always makes the same antibodies. So for example, if I put one right up there, look, that lymphocyte there makes antibodies that look like the one above, kind of Y-shaped ones. But different lymphocytes will make antibodies with different variable groups. So if I've got this one that I've put over there, still making antibodies, but they've got a different shape variable group, a third one up there with a different variable group. All lymphocytes, but 3 different ones, each with a different antibody because it has a different variable group. But notice among all 3 that I've put up there, the constant region remains the same. That's really, really important. Okay, lots of students over the years have tended to confuse antigens and antibodies. Remember, an antigen is just a marker on a cell surface. An antibody is specifically made by a lymphocyte and it binds to the body of a pathogen. The anti-bit, antibody is key on that because it's anti the pathogen, okay, against the pathogen. Now, let's put these into action. Remember, these are still part of the war. So if there's a pathogen, I've just picked one of these pathogens and I've put it up there, look. That pathogen isn't complementary to the antibody on that first lymphocyte. That Y-shaped structure, it doesn't fit that one. Let's put it on the third one there. You can still see it doesn't fit into that variable group. But if we put it on the middle one, you can see that one of the bumps on that pathogen, and we'll say that that bump is the antigen of that pathogen, perfectly fits the antibody shape or the variable part of the antibody on that middle lymphocyte over there. Now, if a lymphocyte locks onto a pathogen, it activates that lymphocyte. And that's actually the proper word for it. That lymphocyte is now activated. When a lymphocyte is activated, it does rapid mitosis and makes loads and loads and loads of identical clone copies of itself. Now, the reason why that's so important is that all of those clones and copies will all be making loads and loads and loads of copies of that antibody, and that antibody we know is specific to the pathogen that we know is in our body and is likely to be in our body in great numbers. So what these antibodies do is they can start to bind to all of those pathogens that are in our body that might be making us ill. Now, you might wonder what the point of that is. Well, some antibodies have the capability of directly killing the pathogens. So just by having an antibody attached to it, some of those pathogens might be killed and then can't do us any more harm. But what's actually better is if you notice on those antibodies over there, they've all got the same constant region. I've highlighted it in yellow on those ones attached to pathogens. That constant region is exactly the same on all antibodies. And there's a reason for that. And it's because it acts like a kind of flashing beacon to attract this guy. Well, this is the phagocyte from the last slide. Phagocytes absolutely love to find, eat and destroy constant regions of antibodies and whatever they're attached to. Now, they can't find the constant region when the antibody is still inside the lymphocyte, look, because the constant region is inside the lymphocyte. But when the antibodies are released, when there's been a response, which you see here, the antibody is now out, exposed in the blood or in the tissue or wherever it is in the body, and that constant region can attract phagocytes, and the phagocyte will very quickly come to it and munch whatever the antibody is attached to. In other words, it dramatically speeds up the speed of response by phagocytes, because those phagocytes, instead of having to go around checking every single cell, they can kind of go, oh, constant regions, go to them, munch them, eat whatever they're attached to. And it really speeds up that phagocyte destruction response. So it leads to a much quicker decrease in pathogen number. Now, we also said, of course, that these pathogens are likely to release toxins. So if I put the toxins on there now as well, you might notice that the toxins, those little stars, are complementary to the lymphocyte up there. Now, exactly the same would happen. If that toxin binds onto that lymphocyte up there, that lymphocyte will activate, will multiply, will produce loads of copies of its own antibodies. So you'll see I've put those in the mix now. And those antibodies will bind to the toxins, again, for the same purpose. They'll attract a phagocyte and the phagocyte will come and eat all the toxins as well. Some antitoxins, just by being bound to the toxin, neutralize the toxin and stop it working. It's like if you had a little bottle of poison, if you wrapped it up in a load of jelly or mud or something, well, you can't get to it, so it can't do any harm anymore. Same if you wrap an antitoxin around the toxin, it stops it working. But the main thing is it attracts these phagocytes. Antibodies as well can help clump the pathogens together in bigger lumps so they can be swallowed in one go by the phagocyte, and it can do phagocytosis of a whole load of pathogens in one go. But the key point is that the phagocyte response is dramatically increased. So the lymphocytes and the phagocytes act as a team to rapidly increase the speed of a response against a significant infection. Now, you will have witnessed this. You just won't have thought about it because you didn't have this knowledge at the time. But I would bet at some point in your life, you've been really ill from some kind of bug and you've been lying there feeling awful. You've maybe been sick, you've got a headache or whatever, and you just felt like you can't do anything. And then suddenly you'll go, oh, you know, I feel a bit better. I might have a piece of toast or I might have a cup of tea. And you do something and then you go, oh, do you know what? I feel a bit better now. I might have a little bit of a walk. And you get better quite suddenly. You go from like, oh, I felt awful this morning, felt like death this morning, to then within a couple of hours, you go, oh, do you know what? I feel much better now. You suddenly feel better. That's because you've had this lymphocyte response. While the pathogen was winning, while it was attacking you and your phagocytes were trying to find it, but slowly, because they've got to check every cell, the pathogen was winning and making you ill. But as soon as the appropriate lymphocyte got stimulated, activated, multiplied, flooded your blood with the antibodies, increased the speed of response, that's where you can get better within a couple of hours. That's why people tend to get really, really quickly better at the end of an infection. Right, take a screenshot of this one because this has got all the information about lymphocytes. We're now going to look at immunity. So far we've looked at how your white blood cells can fight off an infection or try and stop it taking hold in the first place by your phagocytes constantly checking out for them, constantly destroying any that they find. If some do get a foothold in your body and start to multiply, that's when your lymphocytes hopefully get involved, release all their antibodies and fight it off. But immunity is when you can't get a disease again. You quite often hear about, you get a childhood disease, you get chickenpox once, and then you can't get it again. That's not entirely true with chickenpox because there's a few different strains of it. But with a lot of illnesses, once you've had it, you can't get it again. That's what immunity means. Now let's look at something linked to that first, that there are two main ways in which a pathogenic infection can make you feel ill. Firstly, they can either be directly destroying cells, so things like a viral infection, viruses replicate inside your own cells, burst, destroy the cells. The second way they can make you ill is that they're releasing toxins. We've just talked about toxins, but if there's enough pathogens inside you, releasing enough toxins, they're literally poisoning you with their toxins, and that's what can make you feel really, really ill. Now, in both instances, to be ill, it takes a significant number of pathogens to destroy enough cells or to release enough of the toxins to actually make you feel ill. And when I say feel ill, I mean you're displaying symptoms. You've got a headache, you've got vomiting, you've got nausea or whatever. Okay, so you need a lot of them in your body to make you ill. Now, to be ill, the pathogen must be replicating faster than the phagocytes can find it. Because if it was replicate, one phagocyte became two, pathogen became two, but a phagocyte ate one, that one now becomes two and the phagocyte eats one. If they balance out, the phagocyte level stays standard and you don't get ill. If they're replicating quickly though, faster than the phagocytes can deal with them, that's when the numbers can increase. That's when you're going to get ill. That would mean you're losing the battle. So anytime you're ill, you're losing the battle against the pathogens. Now, to win, you need that lymphocyte response. Specifically, we're talking about something called B lymphocytes. You've got two main types of lymphocyte, B lymphocytes and T lymphocytes. At GCSE, we only deal with B lymphocytes. So that's what we're talking about here. If you have that lymphocyte response, so just like this is literally copied from the last slide, the pathogen activates the lymphocyte, it multiplies into big numbers. You've got that army to fight the current battle against the current pathogenic infection, and you can hopefully win. But there's a problem. When you're born, you've got loads and loads of different lymphocytes, but very low numbers of each type, and some of them might be totally unique. Now, I'll try and explain that a bit more. If you think from the last slide, I put 3 lymphocytes up at the top. 3 of them, but they were all different. Now, you need to try and imagine, it's easiest to think of it when you're born. Imagine you've got 1,000 different lymphocytes with 1,000 different types of antibody. But for the sake of argument, imagine you've only got 1 or 2 of each of those types. Very, very low numbers. Now, where that's a problem is that it might take a long while for the specific lymphocyte to encounter its specific pathogen and have a response. You imagine if I've got, I've cut my shoulder and I've got a bacterial infection in my shoulder, but the lymphocyte with an antibody that has a variable group that will fit the bacteria in my shoulder is currently floating around down in my foot. And then it floats around up my leg and it floats around through my intestines and then it floats up down my other arm. It might take a couple of days until that lymphocyte gets to the right place where the bacteria are, the bacteria locks in and it has this response that I've got above me. Now that whole time I'm fighting this infection, I might get seriously ill. Those bacteria might be releasing loads of toxins. It might kill me before my lymphocyte that can deal with that bacteria actually encounters that bacteria. Now that's a big problem. Now immunity gets around that problem. What happens with immunity is that if you assume the lymphocyte response works and you survive, so assuming that lymphocyte did eventually get to my shoulder, multiply, release all the antibodies, lock the antibodies onto all the bacteria, and my phagocytes quickly munched their way through them and made me better, assuming that happened and I survived and I didn't die, then this happens. Some of these cloned lymphocytes remain. Most of them die off over time. So, you know, within a week or two of me getting better from this cut on my shoulder, most of those cloned lymphocytes that could deal with that bacteria are now dead. But some of them remain, and we call those memory cells. Now, they're still there in my body in reasonable number. When I first had the infection, there was that one floating around in my foot initially. After that bacterial infection, there might be hundreds of memory cells still left floating around in my body. Now, what that means is that next time, say I now get a cut on my hand and that same type of bacteria starts to infect there, well, if I've now got 100 memory cells, all clones of the original, all specific to that bacteria that's getting in, well, with 100 of them floating around my body, chances are at least one of them will be somewhere near my hand, will be stimulated almost immediately and activated into having a response, will produce all those antibodies, tagging onto the low levels of bacteria that are in my hand, so the phagocytes can come along, deal with those bacteria before they have a chance to multiply to the levels where they can release enough toxins to actually make me ill. Hopefully that makes sense to you, okay? What it means, that sentence there, with multiple copies of the specific lymphocyte, with the memory cells, the chance of a lymphocyte response to that specific pathogen is now significantly higher and will be faster. In other words, if you've encountered it once, you've got multiple memory cells that remember it, that will be there to deal with it next time before it can make you ill. So this green box really summarizes immunity. You are immune to a pathogen if your body can respond to it and destroy it before it has chance to establish itself enough to make you ill, to get enough numbers to do those things, to damage enough cells or release enough toxins to make you ill. And the only way to do that is if you've got the memory cells in big enough numbers that they can defeat it, tag it with their antibodies, get the phagocytes involved, destroy it before it can make you ill. Now, what that means as well for you is that the battle against that pathogen will have been fought and won without you even knowing it. So any illness that you've had in your life, if it was of one specific strain, you might catch that same illness again, but that second time, your memory cells will recognize it faster because they're in greater numbers, release their antibodies, recruit the phagocytes, deal with it, and you wouldn't even know about it because that pathogen wouldn't have had chance to multiply to the numbers where it can make you ill. So it's not that you can only get a disease once, it's that it can only make you ill once because you can fight the battle much faster the second time round. Hopefully that all makes sense. Take a screenshot of this one, and that's it for this section. So I want you to head now over to disease 6, which is on vaccines. So we just covered the way in which the body can establish natural immunity by actually having a disease, fighting it off, being ill, recovering from it. Vaccines are a way that you can develop immunity artificially by mimicking the same pathogens to create exactly the same immune response to generate memory cells. So head over to that one. Now on that video, we don't just look at the mechanism of vaccines, we look at some of the history, some vaccination programs and the impact that it's had on humankind and our societies. As always, if you haven't done so already, please do subscribe to my YouTube channel. It really helps me make more videos the more subscribers I have. Like this video if you found it useful, and go and find my Facebook page for the Conscientious Biologist. There's loads of interesting infographics on there just to give you a bit more random bits of biology to fill out your knowledge. Thank you.